for the second day. Actually, I was about to start, Margaret, and since I have the pleasure to see you, please come to the podium as soon as you can so that you can share us. Maybe I can, since I'm here, I will introduce Brett, and then you can take over. That's fine. So it's my great pleasure to introduce my co-organizer of this meeting, Brett Finlay. Brett is an old friend, so we've been working on microbial pathogenesis for years, and then we both saw the lights a few years ago, and he's really moved hard into the field of chronic interactions between microbes, the microbiome, and host. And his recent work has really been marked by some pioneer observation with regard to asthma, and it's not just, experiments in mice. It's also a beautiful study in a cohort that is showing some interesting results with regard, again, to the reduction of microbial diversity and the occurrence of these postmodern chronic epidemic diseases like asthma. So, Brett, thank you very much, and the podium is yours.
Thank you, Philippe. Good morning. What I want to do in this talk is to really, we've been hearing about chronic diseases, microbiome and stuff. I kind of want to really drill into that and sort of, we'll use as an example, show you where we stand in terms of what we know about microbiome actually affecting diseases that several years ago we thought they'd have nothing to do with it. So just to remind us all that the microbiome has been around an awful long time. Anthony van Leeuwenhoek really discovered it when he swabbed out his mouth and looked at the animalcules in his mouth. Despite that, we've kind of ignored it up until about the last decade. And part of it is because it's been very difficult to grow. It's anaerobic and stuff. But as you've heard in this last day, that it's really exploding, and really it's a renaissance in the field of microbiology, but also impacting many other fields. And as Philippe said, I've worked on infectious diseases all my life, so I love feces, so that's why this talk is restricted for fecal content. So the joy of talking the second day is you've seen some of these slides, but I'm going to leave them in anyway because I think this is probably my most favorite curve, because this says it all. So yes, infectious diseases have been going down the last 50 years quite nicely, vaccines, antibiotics and stuff, and at least in the developed world, we don't see that much infectious diseases compared to what we used to. And that's been a real success story. But of course, the second half of the curve is really why we're here today. All the diseases that we see in our society today have gone up, whereas these have gone down. And as Marty talked about yesterday, the question is why? And the microbiome seems to be playing a major contributing factor to this. We've heard about the hygiene hypothesis, the disappearing microbe hypothesis. The bottom line is that as we cleaned up our world, got rid of these things, there was a lot of collateral damage to microbes, and we're now starting to realize that somehow, and we're still sorting out all the details, they play roles in these areas here. And asthma is this one here that I'll be talking about today. So, in terms of the microbiome, there's two periods of life where it's really, really important. Early in life and late in life, the bookends of life, if you want to call it that. And really, generally when you're born, that is, your first birthday present of your big fecal bolus of microbes and vaginal microbes, and that really kick-starts it. And for the first year and a half, 2 years, the microbes bounce around, and this is as the kid gets used to it. Then they switch to solid food, then it kind of stabilizes, and it stays that way until then 65 plus, where it starts to shift again, and later in life you see these quite different changes in the microbiome. So it changes here, but what we're going to realize is that this actually plays a big role in the development of us as an organism. So we know the microbes play roles in several things in development. They affect how the gut seals up. There's some interesting data coming out on how they affect the brain develops. But probably the most profound area that we've really come to realize in the last few years is they affect how the immune system develops. And there's many stories that have come out. Whoops. Okay. Thank you. Okay, so immune development is a really important aspect of life, obviously, and really this happens early in life, and we now realize the microbes are really involved in many aspects of immune development, and as you will see that as we get into this talk, that's one of the ways the microbes influence asthma. So just remember, early life microbes affect immune development. So I want to talk about asthma, and the question is, why would I be talking about asthma given I worked on diarrhea? And I'll tell that story in a second. But asthma is one of those diseases we saw on the curve. It's a developed country type disease, and you can see, you saw this curve yesterday. It's going up in most countries. Red is bad. Developed countries have a lot of it. Brazil is interesting because it's probably lower socioeconomical than Canada, the U.S., and Australia, but they have very high cesarean section rates, which, as we'll see, plays roles in there. But really, like all the other diseases we've seen, asthma continues to rise in most of the developed countries. So when you look at what are risk factors for asthma, so it turns out that if you live on a farm, you have 20% less chance of getting asthma. And I really like this picture because I think this kid is grinning because he gets to ride a chicken. I think that'd be lots of fun from a kid's point of view. Okay, living on farm decreases asthma. We heard this yesterday. Being born by a cesarean section as opposed to vaginally, you have a 20% higher rate of getting asthma. Now, that's quite a statement because depending on how you enter this world, that affects a disease that really doesn't kick in until several years later. Early life antibiotics have a similar 20-30% increase in asthma if you have to take antibiotics early in life. And finally, if you breastfeed, you drop asthma rates by about 20% compared to if you bottle feed. So we don't really know what causes asthma. But when you look at all these things here, what are you thinking? Microbes, right? Because that's one thing that all these things have in common is microbial exposure. And the way we got into this whole story is we've been using antibiotics in mice to shift the microbiome and looking at infectious diseases. And one evening I came home and I was talking to my wife, Jane, who's a pediatric infectious diseases doctor. I said, hey, Jane, these antibiotics, they shift, they cause different levels of diarrhea. And she sort of looks at me and says, well, early life antibiotics also affect asthma rates. And it's kind of one of those statements that it just stuck with me. And so it turns out, when you go to the library and look it up, it was true that antibiotics seem to affect asthma rates. But we were looking at gut bugs, and asthma is a lung disease, And so then I started to look for experiments to say, well, how do you know that? How do you prove this? And we couldn't find any. So that's when we said, well, let's roll up our sleeves. We're scientists. Let's do some experiments to see if we can see if microbes are involved in asthma. So the first work we did started with a mouse model of asthma. One of our colleagues down the hall had this routine mouse asthma model. And the way the model works is you take a mouse, and then you take ovalbumin, and you basically immunize it, and then you intranasally challenge it later, and you get this big lung inflammation. This is mouse asthma, and this is a model they routinely use all the time. So we thought, well, let's just take this model, and let's throw in 2 different antibiotics that will have different spectrums of killing, and both these antibiotics don't leave the gut, so will affect the gut microbes, and then let's just run this model. And so we're going to do 2 things. We're going to keep the antibiotics for the whole time, so from very early in life, the whole time of this mouse, or we're just going to add the antibiotics when the mice are adults. So we're going to miss the early life period and see what happens. And, it's one of those moments in science when the experiment actually worked. And it worked beautifully because what happened is that these antibodies, one antibiotic, caused much worse cases of asthma. So I can go through all this data here, but let's just look at the picture. So this is a cross-section of the lungs. Here's the airway in the lungs. This is PBS control, so this is uninflamed lung tissue. You add ovalbumin, so now you get the allergic reaction. See the purple around the airways here? That's lung asthma. That's what people study. Streptomycin looked about the same as this, but when you added vancomycin, you get this horrific inflammation. And the people running this mouse asthma model, they saw this. They'd never seen asthma like this. And all we did was treat this mouse with an antibiotic. So we thought, aha, there's something going on with the microbes. So this is several years ago now when we really didn't have the technologies we do today for characterizing these things. But so each of these vertical lines represents samples from a mouse, and then the colors represent different microbes. And what you can see is when you treat with vancomycin, you drastically shift the microbial composition compared to those that get different antibiotics. So we got to the stage where we had a list of these antibiotics that basically, if you treat with vancomycin, so you're going to cause asthma, these disappeared. So these would be sort of the beneficial ones you might have removed. And then these other 3 microbes were increased, meaning that they might be bad for causing asthma. And we sort of got to this stage and we said, well, what's going on? Why do certain microbes affect it? And then this paper came out by Kenya Honda's group showing that these regulatory T cells, which are a key arm in the immune system for balancing the immune system and would affect asthma outcome, it turns out that 4 of these 7 top here belong to this group. So these 4 were basically associated with how the immune system develops. And they were missing when you basically had this severe asthma. So that led us to chase down what's going on immunologically. And bottom line is these regulatory T cells are known to basically balance the immune system. Th1, Th2 response for non-immunologists. So they're important for balancing it. And if you can't balance it, you can then go towards more of an asthmatic type thing. And so what we found is that in the gut, the regulatory T cells were down, meaning that when you treat with vancomycin, it causes asthma. So basically, this antibiotic seemed to be affecting microbes that affect these T cells, which affects immune balance, and then to do that. But getting back to when this happens, so we had done it, this showed full effects for this. When we treated the adults, I didn't show you the data, there was no effect on the mouse asthma when you treat the adults. So it has to be young mice. So then we did a couple more experiments where we basically just gave the mothers antibiotics and they quit, or we gave them early in life the antibiotics here and then quit, what we call perinatal antibiotics. And what we found is that it turns out that it's in the early time period of life that is when the effect is. The perinatal type here, you can see if you give perinatal vancomycin, you basically really increase the asthma. I'm not going to go through all the details there, but basically that increases here. So it's that really early window in life, while they're still breastfeeding, that it seems to make an effect. So we got to this stage, we said, well, okay, that's nice, that's mouse asthma. What does this really mean in terms of kids? So we then teamed up with a group across Canada that was basically it's called this, Canadian Healthy Infant Longitudinal Development, or the CHILD study. It's basically a birth cohort of a whole bunch of children looking for risks of asthma. So is there a smoker in the house? Were you born by cesarean section? All these types of things. And they basically follow these kids and their mothers from birth until 5 years of age. And they do a bunch of sampling of these things. And so I called them up and says, you guys should be collecting feces. And they say, why would we collect feces? These are kids, this is a lung disease. No, collect feces. So they did, and basically started collecting stool at various time points. And that allowed us then to access feces from these kids at different time periods. So the difficulty is that kids don't get asthma until 3 to 5 years of age. Yet our most data was telling us this is really early in life. It's 3 months of age type thing. So how do we're not going to wait 5 years to figure out what the results tell us. Well, it turns out there's something called an asthma predictive index that basically can tell you what your risk of asthma is by measuring 2 things. One is called atopy, which is basically the skin prick allergic test, and the other is wheezing. And bottom line is if you have both atopy and wheezing, you have at least a 15-fold higher chance of getting asthma by the time you get to school than if you have one or the other or none of those things. So we took 319 of these kids and clustered them into these different groups. The controls didn't have any atopy or wheezing, so these are the controls. 87 had atopy, 136 wheezing. It's these 22 these are really high-risk kids for getting asthma. So these are the ones we were focused on, and we said, well, what's their microbiome doing? So we characterized them on the microbiome, and this is it's called a PCA plot. Basically, the closer the dots are, the more similar they are. You can see the 4 different colors of the groups here. We didn't see any particular, the reds weren't off in one corner, like saying there was difference. Bottom line, what this tells us, there's no obvious differences in the microbiome at a high level when you look at this. So we didn't see any really outstanding differences initially. One thing we did see is that we saw differences in something called short-chain fatty acids. Now, Those in the microbiome field knows these are very important. These are byproducts of fiber digestion, and they signal to the immune system and other things. And what we found is we found a decrease in both acetate and propionate in these kids, in the kids that have atopy and wheezing. So I want you just to tuck that in the back of your mind. We'll get to that in a little bit, what this might mean, that there was these changes in these molecules these microbes make. So we thought, well, okay, if there's no overall gross difference, why don't we dig down and see, are there any little differences in the microbes at this time? So when you start to get down to the family level of microbes, here's your controls. You can see the different kinds of microbes here. And then these are the kids that basically are the high risk for asthma. And what you start to see is way down at this end, there's subtle differences in small numbers of microbes. We thought, aha, there's some differences in these microbes. They're not gross big differences, but there's some nice small ones. So we then dug further, and we basically came up with 4 microbes that at 3 months were down significantly. This is a log scale, it's hard to see, but we're down in all the kids that basically were at high risk for asthma. And here's a controlled bifidobacter was the same. Interestingly, and we kind of expected this given we saw it with the mouse data, by the time a kid is 1 year of age, there's no difference. It's gone by then. So this is, again, early in life. So instead of referring to these by their bacterial names, we basically came up with an acronym we called FLVR flavor, which is basically the first letters of each of these 4 different microbes. So when I say flavor, I'm just talking about these 4 groups of microbes. Again, 1 year, when you look at the differences, there's no difference between these. It's early life, really early life. 1 year is too different. So then we were faced with the problem that most of us in the microbiome face. If you have a disease, the microbes are different than the normal. How do you prove it's actually causing this? There's correlation, but that doesn't mean it's actually doing it. And so we thought about this for a while, and we thought, well, okay, let's see if we can't dream up an experiment that we can actually really test whether these 4 microbes are actually protective for asthma or not. So what we did is we basically took those 4, we grew them up. Now, they're anaerobes. They don't like oxygen. So you have to grow them anaerobically in a hood. Then we took the feces that we had in the freezer from a kid at 3 months of age. We know that had a very high asthma predictive index. He was at risk for it. But also we know that kid actually went on to have asthma later in life. So this is a kid that got asthma. And in that kid's feces, there basically were very low levels of FLVR in the feces. So then we took that kid's feces, and we either took the feces or we took it and added these grown microbes into that feces, and then we put it into a microbe-free, a germ-free mouse. So human feces going to a mouse, plus or minus these 4 microbes. We bred it for a generation, and then to make sure that it actually stayed there, and then we basically put it through the mouse asthma model. So this is a slide just showing that when you add these 4 microbes, so here's the kid's feces. When you add these things, you do shift them, and all these 4 actually are sticking in this mouse. You can see the feces are different there. So that's good. It means they're sticking in them. But what happens when we induce mouse asthma? Well, that's when it got really nice, because it turns out it actually decreased asthma when you put these things in. So you can measure things, cells that are infiltrating into the lungs, and you can see that when you add flavor, you basically decrease the infiltration in here, you decrease the inflammation, and you don't see the heavy purple compared to what you see here. So this is as close as we've got to sort of proving these things are involved. They basically decrease significantly the asthma that you see in the mouse asthma model with the human feces going into the mouse. So then we knew in the mouse it was working through the immune system, so we actually then measured several immune parameters, cytokines that are going on in these things. And what we found, when you add the flavor microbes, you decrease, these are inflammatory cytokines, they go down, as you'd expect here, and significant changes. So basically it's dampening the inflammation in the lungs. That was nice to see. Another measure of asthma is the production of IgG2A and immunoglobulin that's characteristic of these things. And again, you see a decrease when you added these 4 microbes in this humanized mouse-type model. So the way we think it works is these microbes are basically affecting how the immune system balances early in life, and then you basically go towards an asthmatic or non-asthmatic type immune response. So, as always, when you find these things, this is a little scary because here we're saying that fecal microbes affect a lung disease, asthma, which happens later in life. We've now gone on to look at a whole bunch more children, and basically it's holding. Here's the F and the L. These are the most significant ones. They seem to hold quite nicely. You can see they're really decreased in a large number of children in the high-risk group here and here. And as Marty alluded to yesterday, the joy of science, if someone reproduces it, it's exciting. I was very happy when Marty told us as a result that they found the same kind of microbes in their thing. And now there's been several studies come out since we first published this, basically saying that there's correlations with these microbes. Not always perfect, but many studies are starting to show that these early life microbes are really important. Okay, so that's Canadian kids. What about the rest of the world? So we thought, well, let's do a similar experiment in a set of microbes that are elsewhere in the world. And it turns out we went to Ecuador to do this, mainly because I knew this guy named Phil Cooper, who I'd collaborated before with on a Gates grant we had. And he had a cohort of kids that are in rural Ecuador, way up here. And the thing about Ecuador is that its asthma rates, I think it's around 10% or so, they're very similar to the Canadian kids' rates in terms of the asthma. But these are obviously probably very different microbes in these kids because rural Ecuador is quite a different place than Vancouver or Toronto. So we set up to do the same kind of experiment, do the Canadian kids, but with this rural Ecuador cohort. So we had close to 100 kids, and again, the high-risk kids, they have atopy and wheezing versus the controls. And we then went on to measure various things. And you can see that this is basically how many kids actually go on to get asthma at 5 years of age. You can see that the kids in the high risk, nearly half of them go on to get asthma, whereas the controls, very few of them go on. And these are just measures of asthma that the AW definitely have markers at 5 years of age. So for the anthropologists in the audience, wake up for this slide, because this, I think, will appeal to you. So these are risk factors associated with asthma in the Ecuador cohort. And most of them are what I've already told you with what's pretty much known. So if the mother takes antibiotics during pregnancy, you have a much higher risk of getting asthma. If you're born vaginally, you have a lower risk of getting asthma than C-section, of course. Breastfeeding is protective. Ecuador has worms. We know that some worms are protective of other. Make asthma, we already knew this. If you have a respiratory infection, we knew this too, you're high risk for asthma. But the kicker to this slide is this last line. The highest risk for getting asthma was clean, potable water. You give these kids clean water, they basically have a higher risk of getting asthma than if the mother takes antibiotics. So I think this is just the perfect example of the hygiene hypothesis. Here we are, we want to clean the world up, get rid of all these nasty microbes, yet ironically, you're putting them at risk for potential asthma. So we did the same kind of experiments looking for microbes. This is the bacteria we did, and you can see differences in these things. You can find some that are higher risk, some are lower risk. They're different microbes than we saw in the Canadian kids. We kind of expected that because these kids' microbes are very different. So we didn't see the same ones as the Canadian kids. But where we really started, we also, by then the technology had changed so we could actually also look at fungi. So instead of bacteria, we're looking at fungi, eukaryotes. And it turns out this Pichia, this type of fungi here, it turned out it was a huge risk factor for getting asthma. It was higher than any of the bacterial ones we got here. So you can see Pichia is blue here. You can see in the high-risk kids, there's this big blue bar here that's basically non-existent in the controls. So there was this yeast factor associated with it. Again, here you can see the Pichia increase here. So that was interesting, and that led us on a chase. Well, why does having this yeast make you more high risk for getting asthma? So we set up a bunch of experiments to see, well, what is this yeast doing in terms of affecting these kids here? So basically we wanted to see, is it affecting the immune responses in these things? So we could take these mice and then give them Pichia and then basically measure the whole asthma type exposure. And what we found is that if you expose these mice to Pichia, they have this has got inflammation. These are immune cells involved in inflammation. They have a lot more inflammation, which makes sense because inflammation is then triggering it. So then we set up another commonly used asthma model in mice, and this is where you take house dust mites and you take pieces of them, and then you immunize them again, and then you challenge them with this house dust mite model. And this is thought to be sort of an even better mouse model than the ovalbumin model for this. And you can do various readouts to see what's happening in here. So very happily, I guess from a scientific point of view, is that we found that, like we saw in the mouse models with the ovalbumin, is this pichia basically was increasing Th2 cells, which are involved in triggering inflammation in the lungs and basically the asthma associated in these mice. You see an increase in Th2 cells, you see eosinophils, which are also a marker of asthma coming in. So we also saw changes in Th17 cells in the lungs, because that's where the real inflammation is happening, not just the gut. Again, you can see increase in these things here. So we then had a look, does it affect various other cytokines? And yes, indeed, after this house dust mite stimulation, these are inflammatory cytokines. We'd hope they go up and peak affecting it. And yes, indeed, this is true. So then let's get back to the idea of when do you have to expose them to this to affect the outcome. So we did an experiment where instead of exposing early in life, we exposed pichia later. So we added the pichia to these things later to see whether it would actually have an effect. And we saw a big effect in these when you added pichia earlier, but if you added the adult mice, it's done. It's too late. The immune system is developed, and we don't see an effect of this pichia when you add it later in terms of these things. So you remember a while ago I talked about short-chain fatty acid acetate being affected in the Canadian kid cohort. It was decreased in the asthmatic ones. Of all the metabolites we measured, both in the Canadian cohort and in the Ecuador cohort, acetate was the only one that showed a nice correlation. Also in Ecuador cohort, there was a drop in the acetate levels in these things. So that was nice because it said there's something in common at least, and acetate is known to influence how the immune system develops. So it's starting to make sense there. So we then started to say, well, could acetate being affecting pichia? Is there a relationship between this yeast and the presence of this acetate? And it turns out that acetate, which is present is not present when you get asthma, and it's basically protective. It turns out that acetate actually inhibits the growth of this pichia. So the idea being, if you have microbes that are producing this acetate, maybe you're sort of killing off the pichia, or it can't grow or something, so it can't then start to trigger these things. And this is what this is showing here, that the acetate is affecting it. So we're still working away and trying to define how all this fits together and look at the interface of this pichia. Canadian kids don't have pichia. It's just not in their kids' microbiome. But at least in Ecuador, maybe the presence of these microbes is affecting the presence of this yeast, which then is affecting immune development and the outcome. So that's kind of where we stand with this story as of today, that I think that the real concept that there's really an early critical window. People were talking about 1,000 days in life yesterday. I'd say no, it's 100 days or less, 3 months or less. And a lot of the other studies, even at 1 month, it's that real initial bolus of microbes that's setting the immune system up, that's where it's really critical in terms of this. So, and we're seeing this again and again, food allergies, obesity, other things. This seems to be a really sensitive time in a human's life where these microbes really do seem to play a big effect. So, Marty plugged his book. This is one that Claire and I wrote. Claire did a lot of the asthma work here, and this is sort of just, again, our attempt to convince the lay people that we have been in our society now. I think we're being hyper-hygiene, and we got to ease off on this. And these microbes do play a big role in the kids. So with that, I'll say thank you. The work was started by Shannon Russell, who did the mouse work. Most of the cohort studies were done by Claire Arietta, who's now running own lab at the University of Calgary. Rosalind and Therese were doing the pichia and the immunology. And this is done in collaboration with Stuart Turvey, who had the kids cohort, and Phil Cooper, who had the cohort in Ecuador. Kelly is our mouse asthma person, and then our metabolomics were done here. So thank you very much.